All right, hello biologists. We are here talking about learning targets 1, 2, and 3 for cell structure. So we're going to talk a little bit first about microscopes. This first slide shows you a picture of the history of the development of the microscope over the years. You can see early on kind of very simple structures that just have your lenses. You're going to have an ocular lens, which is going to be the one that's near your eye, and then you're going to have a compound lens, or objective lens, which is going to be right here, that makes up your compound light microscope. Okay, I would say the ones that are most familiar and what you've probably seen in classrooms over the years is something similar to G. That's kind of what ours look like, although this one has 2 ocular lenses, so you look through both eyes. We have a few of those, but most of ours you just look through one lens. Again, that's the ocular lens, and then your objective lenses, this one has several. Ours have 3, and your ocular lens is usually going to magnify 10 times, and then depending on the magnification of the objective lens, I think we have 10, I know we have 40, so our maximum magnification is about 400 for our light microscopes. So the light microscope developed, we're talking about way back in the 1600s, 1700s, 1800s. Obviously, until that time, we didn't know about cells. No one had seen them. We had no idea what matter or tissues or any plants, animals, anything was made up of. Those first early microscopes just enlarged things, but they didn't really produce really good, clear images like we have today. So again, that's another thing that technology has advanced our understanding of a lot of things just because we're able to see what's going on. Light microscopes can magnify about up to 1,500 times the actual size. That's the max that a light microscope will get to. Our light microscopes don't go that high. Again, ours max is 400, but about the max for light microscopes is 1,500 times. And a lot of times you need to stain your sample so that you can see the details. So a lot of times it will appear to be colored, like a cell will be colored, but in actuality that's just the dye that's used or the stain that's used so that you can see it. The other type of microscope that you can use to study the details of things are electron microscopes, and there's actually 2 types of electron microscopes. We have, well, first of all, before I get into that, what electron microscopes use, obviously, is electrons. I mean, we named the light microscope a light microscope because we use light and lenses. Here, we use beams of electrons, and the magnification power of electron microscopes is much higher, about 100,000 times. We don't have light microscopes at Rosemount High School, but most likely the college that you go to, they will have electron microscopes, and you'll be able to use those if you take biology classes. And these, they have to be studied in a vacuum. The 2 types of electron microscopes are TEM microscopes and SEM microscopes. Scanning electron microscopes, that's what the S stands for, and then TEM is a scanning tunneling, so the T comes from the tunneling part. I always remember the TEM. I remember the word cut because it has that word T in it, and what a TEM is giving you is a cross section. So if you were to take like an apple that was just more of an oval shape, or a plum or something that looks like that, and you were to cut it in half, a loaf of bread, that works, a loaf of bread, and cut that in half, and you were to look at each side, that is a cross section. An SEM, a scanning electron microscope, is going to give you more of a 3D image. You're going to be able to see detail and kind of a 3D structure. So both of these over here are actually SEM more of a three-dimensional type look. This is blood, so you can see red blood cells in here. You can see some phagocytes. You can see all kinds of things, that platelets that make up our blood. And then here are some what looks like bacteria cells that are undergoing division. The book has a page that has these pictures on it. Again, here is your SEM. This is actually showing the stomata. This is a plant cell surface. Plant cells have little openings that are called stomata that allow for the exchange of gases. This is a TEM of that same structure. That's a bad arrow. I'll make it colored in. There we go. That's still bad. That looks like a triangle. Anyways, this entire thing is your TEM. This got cut off for some reason. Your TEM, there is your cross section. So if we were to take that stomata, cut it in half, that is just the tissue layers and what you can see on that thin little cross section. And then this is a light microscope. LM is going to be light microscope. And this one is zoomed in 2,000 times. We also talk a little bit about medical imagery. X-rays, we know all of us probably have X-rays done at some point. They're going to pass through soft tissue, so you're not going to see skin and muscle, but those X-rays are absorbed by bone and teeth, so they're great for diagnosing any type of fracture, break of an ankle. Here you can see there are some devices. This person has had what looks like an ankle replacement. The new ankle joint will maintain her mobility much after previous fusion. So there is a ankle replacement. Remember, Mr. Bender is going in Tuesday for his full knee replacement, so he'll have, next time he has X-rays, if he ever needs to have X-rays on his knee joint again, he'll see the metal and titanium that is being put into his knee. And then this is an X-ray showing a pacemaker connected to the heart. Some of you may have also had MRIs along the way. MRI stands for magnetic resonance imaging. It uses magnetic forces to produce a cross-section. And this is great for looking at more of your ligaments, more of soft tissue type injuries. So here's something to think about. Again, here is our two types. We have our MRI over here. We have our X-ray over here. And I guess the question will be, why would a doctor use an X-ray over an MRI, or vice versa? CT scans are another form of medical imagery. They're computerized tomography. So it combines a series of X-ray views taken from many different angles. And you can kind of put together a picture of an organ and see what's going on. So again, your doctor would have to choose, why would we do a CT scan over an MRI? Well, it's going to depend on what needs to be seen and the reason for the exam. CT scans usually are good for seeing and diagnosing cancer, pneumonia, chest X-rays, any brain bleeding, or things in the lungs, like again, pneumonia, looking for that, bronchitis, things like that. MRI is better for seeing ligaments, tendons, the spinal cord. So most people, if they have some sort of sports injury, you know, Mr. Bender had an MRI for his knee to make sure it was just the bone. He had X-rays as well, but looking at the tendons to make sure all those things were intact, and it was just the bone that was causing the problem. Oh, and look who we have here. Oh, and you heard her too. Roxy is in the house. We are sitting on the couch working on the screencast. She wanted to pop her little head in and say hi. I actually tried to get her to sit next to me, but she's kind of lazy right now. She needs a nap. So she's laying, and I have the iPad taking it from above. So look at her cute little nose. So Roxy's in the house. She says hey. All right, couple things to finish up. Learning targets 1, 2, and 3. The cell theory. The cell theory was, we'll talk about the scientists that were crucial and instrumental in developing this theory. But remember, back in the 1600s, 1700s, they didn't even know that cells existed. So this theory may seem today like, well, duh, we know there's cells. We know everything is made up of cells. But this was really revolutionary at the time when cells were just being discovered. So there are 3 parts to the cell theory. First part is all life forms are made up of 1 or more cells. So any living thing is made up of cells. That is, a cell is the basic unit of living things. And it has to have either 1 or more cells. Cells only come from other cells, so they just don't appear out of nowhere. We have a process called cell division, which we're going to look at. So I guess the most basic form of cell division is when you talk about development, you have your egg cell and your sperm cell, which come together to form your first cell for your new baby. And we'll talk a lot about development, meiosis, cell division, those early cell division. And then the third component of that is the cell is the smallest form of life. So again, the building block of life. These scientists were instrumental in contributing to the cell theory, and you're going to need to know their names and what their contribution was. So I'm just going to run through them quick. And in class, we're going to work on a way to help you remember their names and to keep their discoveries straight. So first off, we start with Hooke. He was in the 1600s, 1665. He was actually the first to coin the term and use the term cell. So he observed cork and saw these tiny hollow little compartments, and it reminded him of the cells that the monks in the monastery used to live in. So he coined that term and said, oh, those just look like those cells, so we're going to go with cell as the name. And it has stuck since 1665. Next, we have Leeuwenhoek. He was in 1674, very similar time frame to Hooke. He used a single lens microscope and observed pond water, and he saw these little living life forms in pond water, which today we know, yep, duh, we know there's things in the pond. He called them animalcules. So not molecules, but animalcules. So living things that were just small little structures. In 1838, now some time period has passed, the microscope has been useful in looking at more detail, still kind of crude detail, but nonetheless, they were able to see them. Schleiden, he proposed that plants are made up of cells. And not long after, a year after, Schwann said, okay, we know that all plants are made up of cells, all animals are also made up of cells. And then our last individual was Virchow. Excuse me. Sorry. In 1855, he said all cells come from pre-existing cells. So that was that last part of the cell theory. So main things, you know, the cell theory states that all living things are made up of cells, plants, animals, and all cells come from pre-existing cells, and cells are the basic unit of life. We're going to study a lot about cells, different shapes, sizes, functions. We'll get into all of that, but you can see we have a, probably if you've, drawn cells before in elementary school, middle school, seen drawings, you've kind of seen probably your basic animal cell structure, which kind of looks a little bit circular, but cells come in all shapes and sizes. We have these super specialized cells of our nervous system. We have red blood cells, which are specialized for carrying oxygen. We have muscle cells, which we know help contract and move our bones. So we'll look at a lot of these different types and then how they get to be different than that first cell, which came from the egg cell and the sperm cell. That's fascinating as well. It's called cell differentiation. And then to finish up this screencast, the difference between prokaryotic cells and eukaryotic cells, I'm going to hit this real quick because we're going to spend more time next week talking about it. But prokaryotic cells are going to be your very simple cells. All right? They have no nucleus. They have no what are called organelles, no membrane-bound organelles, and they are most of them are single-celled organisms. So bacteria are your example of prokaryotic cells. Eukaryotic cells, on the other hand, are going to be more complex. They do have a nucleus, and we know that one of those organic compound groups is found in the nucleus. Think back, quiz yourself. What's found in the nucleus, or what stores our genetic information? That would be the nucleic acids, DNA and RNA. DNA is found in the nucleus. Organelles, all of these little tiny organs, that's what organelle actually means. We have organs like our heart and our lungs and our kidneys and our liver and all that stuff that does jobs for us. Well, these organelles are tiny little organs that do similar functions in our cells. Okay? Eukaryotic cells can be just one cell. There's a specific type called an amoeba. Oh, am I going to spell this right? Amoeba. That are just one single cell, but are very complex. They have organelles. They have a nucleus. And then multicellular as well. Most eukaryotic cells are part of a multicellular being like us. Okay? We have eukaryotic cells. So this includes all of our plants and all of our animals. All right? And we'll talk a lot more in class about organelles, plants, and animals. I couldn't resist putting one more picture of Roxy in the screencast. Also, remember to bring your pictures of your smiling pets. I want to see your dogs and hamsters and cats and all your pets. So that is another homework assignment for the weekend. Bring those in. I've gotten a few, but I want a lot more. Roxy says bye. Good girl, Rox. Yeah, good girl.